Do you need to start the recording? Oh, okay. Great. All right. Well, thank you, everybody. I'm going to talk a little bit about what do you want to learn. I'm anticipating that my portion will take around 20 minutes, and then we'll pause for questions, and then move on to Chris's talk, which will go into a little bit more of the nuts and bolts of designs. Feel free to put things in the chat. I might see it as I go, but more or less, we're going to try to focus on getting through our planned content and then branching out to your specific questions. I do want to acknowledge the funding for the course over the years. This was previously funded under an R25 back a number of years ago, and then moved on to Chris's infrastructure grant for the Data Coordinating Center for NeuroNext at Iowa, and now has moved to the University of Virginia and Indiana University under Drs. Quigg and Gutman. We have had gracious support of the American Academy of Neurology as well for some of our activities. Here are some disclosures. Sorry, these are popping out a little slowly. I do episodic medical legal consulting. I do some consulting for Berry Consultants. I work as a reviewer for PCORI and NIH. I do get paid to do methodology statistics reviews for a couple of emergency medicine journals. And I do have some funding from the NIH to do a few clinical trials in vertigo, hypertension, and adult and pediatric cardiac arrest. So I think it's helpful to orient ourselves as to what is the purpose of a clinical trial and why are we trialists. It's also consider what sort of goals clinical trials should have and what those look like, to give you better clarity when you're sort of dedicating part of your career towards discovery, and understanding what you can get out of a clinical trial. What's the output, the deliverable, so to speak, to society and the world, and then how you update your path based on the data that you gather from a clinical trial. So I will pause at each of these section headers where we talk about, sort of 4 different broad concepts. And again, if you have some things in the comments, I'll take a pause and try to get back to things that have come up in the last little section. I've used these pictograms to determine whether I'm describing a pearl or a pitfall. Because I'm reasonably old, I really enjoyed playing Pitfall, the video game by Atari, back in the day. So that is the icon for that. And you can see the other one is a lady clutching her pearls. So what are you going to be taking away from this after you hear from me and Chris? I think getting the question right is essential, and we spend a lot of time on this in the course. We spend a lot of time on this as we're selecting people who are good candidates for the course, because if you're not asking the right question, you're potentially wasting your time or wasting the time of research participants or wasting resources of others. You have to be a little bit cautious about myths that sometimes you will hear from people, like you always have to start with safety and prove safety first. Now, you certainly don't want to start with dangerous things, but safety can be a challenging thing to establish. It's really more or less establishing a lack of unsafety, depending on the scenario. A useful thought experiment is for you to fast forward yourself to the end of the trial and imagine what its results are and what people are to be doing next with those results. You should potentially ask yourself if what you're doing is an underpowered efficacy trial. If somebody suggested that to you, like, just see if there's a whiff of an effect, so that is promising to ask for an investment in a larger trial. That's a form of a sort of a statistical lottery, and is often done, but is not a good idea, and is definitely discouraged by funding agencies and other scientists. And you're trying, like science involves the potential to prove or disprove something. So if you're doing a clinical trial and there is no chance it's going to give you information that tells you need to change what you're doing in the next phase or stop what you're doing altogether because it's not going to work, then you may really need to go back to your design and think about whether you're getting enough out of that design. Now, that doesn't mean that sometimes you may not have goals that pertain to feasibility and so forth. That is completely an option, and it goes back to that top point of getting the question right. But you have to have some bar to say that it wasn't feasible, right? There needs to be some possibility that your trial can tell you to stop or change what you're doing. If you already know what your trial is going to say, then you're missing an opportunity to do something to learn. So in terms of the prologue, I think for you as professionals, this is not only a course in scientific design, it's a course in professional development. Some of this is something that we all need to do, even those of us who are more senior, is thinking about what is our vision for ourselves and how we can create a vision of greatness for ourselves. The idea and book of creating a vision for greatness is something that was written by Ari Weinswig. If you're familiar with Ann Arbor or been there before, he is the founder of a famous deli, Zingerman's, which also has a lot of training courses and has a really impressive way that they take care of their employees and listen to their teams. So thinking about how to do visioning is something that is a useful exercise. And I think it applies to you as you are imagining what your career is going to take, what twists and turns it will take, but also as you think about visioning, what is going to happen at the end of this trial that you're designing? What are the results going to tell you and the community needs to happen next? So thinking about this, there are these two tracks. What information is the trial going to provide? How will it move science forward? And what information will the trial provide to move your career forward? I think those are important. And sometimes you can be very clever and really design a trial where the answer is interesting no matter what, even though it may tell you that a clinical trial isn't the next best right step. It may tell you something about the natural history of the disease or other characteristics of the patients that will give you really important knowledge for the future, or what information it will give for your career. Sometimes senior people talk a lot about their successes and maybe not enough about their things that were, we can use the term failure, a clinical trial. Like I have an example. I came up with a really elegant clinical trial exam when I was doing a master's degree in clinical research design and statistical analysis to look at, trying to blunt the neurocardiogenic injury of the heart after subarachnoid hemorrhage with early beta blockers. It was a wonderful trial. It was beautiful. It involved before and after echocardiograms that were getting quantitative measures of the heart. I even got a secondary study to look at you do a nuclear medicine study of the innervation of the heart. And the study was completely impractical. There was no way to get an echo promptly that could give you quantitative measures. There was no way to move a really sick subarachnoid hemorrhage patient down to nuclear medicine. But even though I did not enroll anybody in that trial, I learned a lot from the experience. And that was information that helped. It didn't tell me that I should just run away screaming from clinical trials. It just made me think more about how you frame the question and how you think about what will work. So the output of the trial, will the data change your future actions? So if you're looking at what your trial is going to tell you as you plan it, if the answer is no, again, reassess the design. And this applies both to, will it change sort of the pathway of your career, or will it change the pathway of the trial? Or the next trial, so to speak. So part one, the scope of the problem. You're here because clinical trials are relatively hard to do, and we have a lot of experienced faculty members that can help you sort of, sorry, like, 1 second. I just have to like, I'll be right back. Hey, Liz, I think they're here. All right, sorry about that. The doorbell doesn't work reliably, and I was in charge of ordering pizza, and it's 16 minutes early, so sorry. And if you can, finish your clinical trials early too. So that's helpful. All right, so why do clinical trials stink? Number one, you can think about it, particularly for many of you who are clinicians and clinical practitioners of medicine. A clinical trial, in certain respects, is like a diagnostic test. You can use this as an analogy, and my analogy does not stink. But if you think about the diagnostic test, you have your sensitivity or your true positive rate, and then you also have your false positive rate or 1 minus specificity. And when you're putting together a clinical trial, those things are power and your significance level. And the way to think about this is that your clinical trial is screening for treatments and some combination of a treatment, a population, inclusion criteria, and a delivery method, and an outcome measure that will allow you to improve patient outcomes. You're going to learn about a new treatment and who and how to use it in. And that if your trial can find that combination of things, then you will have a new treatment for that group of people. And that is why this is one way of thinking about this is a diagnostic test, right? It's like, when we use it, it's like, is this patient likely to have this disease? Is this a treatment that is likely to be efficacious? And when we make a clinical trial design at the beginning, we do have to make lots of assumptions. Sometimes we have to think that animal models might give us close to the right dose. That sometimes we say there's no heterogeneity of effect because it would blow up our trial if we allow it to be somewhat robust to that assumption. Subgroups that will respond equally. We will exclude some subgroups because maybe we think they won't respond, or they'll have too poor a prognosis or too good a prognosis. We sometimes we will search for an effect that we know is feasible because we know that perhaps our funders will only fund trials of certain sizes. Can we understand the noise and the outcomes? And if we don't know enough about the noise, then it's hard to understand how big our trial needs to be to overcome that noise. Are we coming up with a practical treatment plan? And it's hard to get good quantitative, unbiased information on all of these things. So we do have to make a bunch of compromises to reduce this to make a model solvable. But the more that you think about which of these decisions that you're making explicitly can be very useful. But yeah, you can't necessarily design a trial design where you will reduce the uncertainty in all of these things. But if there is something that is very important and has important uncertainty, it can be a real important option. So what are some good questions for trials? And Chris will go into this in a little bit more depth, but you should have one main question. Is X better than Y? Or does X improve a patient-oriented outcome compared to Y when administered to this population for this long at this dose? So your study should have one main question that goes back to that. What do you do next? And can you make it an even better question? What dose, schedule, and population would hold the most promise for determining that treatment X can be proven to be better than treatment Y in a phase 3 trial of size N? So that's a different way of looking at what a phase 2 trial might, or an exploratory phase trial might tell you. So you might be asking a really good question, but sometimes you may be asking too many questions at once. So if you do get the question wrong, there are risks, and this is the pitfall. FDA is very concerned about falsely declaring an ineffective drug efficacious, because then there's something out there on the market that we're spending money on that isn't helping patients. It's not likely to necessarily be harmful, but if it's neutral, that's a big problem. On the other hand, we as scientists and academics need to worry about falsely declaring an effective treatment neutral because something was wrong in the clinical trial, whether it was the wrong population, the wrong dose, the wrong treatment window, the wrong schedule. If we design our trials poorly, we may bias the world against a treatment that could have been effective if we looked at it the same way. A great example of this, although we did thankfully keep looking for better ways of doing things, would be endovascular treatment for stroke. We had a number of neutral trials, but something was wrong with those trials in terms of how long they were taking, how much the tech was changing, under background care, and when those patients were being enrolled, and in fact, whether those patients even had large vessel occlusion. So all of those things were reasons for those earlier neutral trials. They fortunately didn't dissuade the world from continuing to do trials, but it could have gone a different way. So think about that. If you have a problem in your trial design, you sort of bias the world against something that might have been effective had it been given a better chance. It's hard to think about what outcomes need to be measured in an early phase trial. Considering the likelihood of effect on an outcome versus clinical importance that is patient-centered. Timing of follow-up can be a major practical issue. Definitely the reliability and prior validation or use of the outcome measure is crucial. You can consider having multiple outcomes, but again, there's a risk of a false positive result due to cherry picking. And your trial design, is likely going to collect, multiple outcomes, but I think you have to be very discriminating and cautious about how you choose those, and be very transparent and explicit about what is the main outcome. And again, the main outcome might be something that isn't a patient-oriented outcome. It may be something that pertains to feasibility. Thinking about the disease phenotype is important, considering where you might be able to find patients, what types of confounding factors and treatments, and what kind of established outcome measures. Sometimes you might need to use the same agent in different diseases. Sometimes something that can be done, although in the sort of early phase investigator-initiated space, it would be a little unusual, is something an indication finding trial. If you're kind of looking across a variety of different diseases that are related and or phenotypes, trying to leverage that investment and find out who is most likely to respond to a treatment so you can help create an indication for that. There's always a tension between enrolling a narrow population and a broad population. The good thing about narrow populations is there may be less variability and easier to interpret results, but it will take longer to enroll patients because you have to be more selective. There was some examples of this in some of the extended window thrombectomy trials where they had very large treatment effects because they were very discriminating against patients who were really likely to respond. And in the subsequent years, we've worked to try to expand those indications to more and more patients. If you start out with a broad population, though, you may have too much variability to actually see the treatment effect because it's going to be washed out by a lot of those patients who are having a less substantial response to the treatment. To inform the design of a confirmed phase trial, you do need to enroll a population that's similar. It may be broader, and typically it shouldn't necessarily be narrower in your phase trial. So the epilogue here, and then I will hand things to Chris, and if you have any questions, please pop them in the chat, is one thing is you design a trial. It can be very helpful to think about the consort and spirit checklists. They have extensions for a variety of different trial designs, including pilot and feasibility trials, multi-arm trials, non-pharmacologic interventions. So going to consortspirit.org and looking at the statement. Now, this is for publication. It's a checklist for publication, but it can be very helpful for you at the design phase as well, because you should think about the issues that they would be asking about when you submit your manuscript as you're planning your trial. So I find that this can be quite helpful. And reiterating the takeaways from me, getting the question right is essential. But beware of the myths, like always start with safety. Again, safety may be important to evaluate, but if you're only looking at 20 patients and the expected rate of your unsafety, your safety event is 5%, you're at most going to observe, you're more likely than not going to observe 0, 1, or 2 of those in your trial. So you're not going to gain a lot of information. So think about whether there's something that's a more logical main outcome, or is it good enough to just say, as long as there's fewer than 3 safety events, we've confirmed that there's a high likelihood that this safety event is at a roughly 5% rate. Again, you're not going to prove safety, but you can establish that there is a lack of unsafety to some margin. Fast forward to the trial and think about what it's saying and how you'll use those results and how you're going to communicate those results and what you're going to do next in your career and for the next trial. And always ask yourself if it's an underpowered efficacy trial, and pivot or changing your approach to the next trial should always be an option. With that, I will unshare so Chris can start to get his slides ready. And Chris, it appears I've stunned them into silence, so they're really ready for you.
All right, well, I'll just pick up and then we can take questions at the end, cover them both. So I'm going to kind of take a lot of what Will said and reiterate it, but whereas Will kind of covered it from more of a clinical perspective, I'm going to cover it more from a statistical perspective. This kind of gets to the key point that Will was hitting at, but I think it lays everything in terms of the basics you have to start with. Each clinical trial has to have one or more primary objectives, which are basically the question or key questions. Usually there should be one high-level question at least. That is the question that the investigators, the sponsors are most interested in answering. And that kind of sets the framework for everything that you need to do for the study with respect to sample size, with respect to the design and so forth in order to answer that question. And having that question is important in terms of the design. And this is, I think, an important concept that will come up a lot when we're talking about design of clinical trials and kind of in the world we live. A negative trial is not one that fails to show that an intervention is effective. A negative trial would be one that fails to answer the question. So if you design a trial, if you're asking a question and you have equipoise, at which you should in the clinical trial, if you can set up a design that kind of definitively tells you this is worth moving forward or this is not worth moving forward, that's a success from the trial design framework. It might not be from the person who was hoping the intervention would work, but if you can answer the question, that's a win. If you get to the end of the study and your design was flawed in such a way that you can't tell from your data whether you've addressed the question in one direction or the other, that's what I would term a negative clinical trial. And I'm pretty sure Will and others would agree with that concept. Will mentioned the research question and gave some examples. I've got on here just kind of poor question, not in the sense that it's not a bad question. Is treatment A better than standard of care? But it's not a question that enables you to design a clinical trial around. And so you have to get into what population, what dose of treatment, reducing what over what period of time. So there's lots of different components that have to be fleshed out. And a lot of the early experience that I've had in clinical trials is you often have the clinician and the statistician who get together and are really coming at that in some ways speaking two different languages and trying to find common ground that gives you something that is addressable and testable in a clinical trial. So the clinician has what they want to do clinically, and then the statistician has to find the common ground with them to put that clinical question into a question that can have a hypothesis and a design generated around it to give you a testable question to try to address is what you hope to achieve, achievable within the design. There are lots of secondary objectives. I just put this in here in the sense that the secondary objectives can be important in a trial, but they can also be red herrings if you're not careful. They can add a lot of cost and complexity to the study. If you end up with lots of endpoints and lots of times on participants, it might be that your number of secondary objectives actually make it harder to get your primary endpoint if people can't do everything. So you want to think carefully about secondary objectives. I've seen protocols where there are up to like 50 secondary objectives, which are probably a bit too much, and most of them are going to be underpowered. So I just put this on here mostly, particularly for the students in the course this year, as you think about your protocol, think about is something like it's really the critical secondary questions that should be secondary objectives, not everything that you think, could possibly be asked in the study, although there are exceptions, of course, to every rule. I'm going to focus more on the primary endpoint, where each trial objective needs to be linked to a primary endpoint that will give you what you need to answer that question of interest. And this is where I meant by the question drives everything. You want to put a lot of care in choosing the endpoint. And in general, the endpoint should be something that matters to a patient. Language used by the FDA typically is something that measures how a patient feels, functions, or survives. So some way to know if you can change this endpoint, that it will have impact on the patients, that, if you reduce it or increase it in whichever direction makes sense, a patient would say, yes, that matters to me. That's of a meaningful scale that would be important for me in terms of my disease progression with the study. It helps to have endpoints where preliminary data are available. You may not always find yourself in that condition. That will complicate things. In our webinar next week, Sharon Yates will talk about sample size. Without any preliminary data, the sample size calculation gets much more complicated because it's going to be based on a lot of guesses that don't have data to support them, and you're going to have to defend it more. So you'd like to ideally use an endpoint where you have some sense of what to expect, either from prior studies or from natural history studies in your disease, to inform both your sample size calculation and kind of your expectation for how it should perform in the study. There is a difference between outcomes and endpoints. I think these two often get conflated, whereas the outcome is the measured variable. So that's what you're actually measuring. And the endpoint is how you would convert that measurement to something that will be analyzable over time. So for instance, for a simple pain study, the outcome could be the measured variability of the patient's pain on a 10-point visual analog scale. But your outcome may be the change over time, over the change in the pain intensity over 90 days. So there is some relationship between outcomes and endpoints, but it's important in fleshing out your protocol and in thinking about this: what is your outcome and what is your endpoint? The outcome will be what you measure, but the endpoint is really what's going to drive your sample size calculation and its assumptions about the endpoint that are going to be critical for that. I just put a few examples on here, and I think these slides we can make available on that. But the outcome, again, so for instance, for stroke, the outcome may be occurrence of stroke, but the endpoint might be a reduction in the proportion of patients with stroke over 3 years. So what you're going to use to analyze that, and I've given several different examples for a number of different neurology examples here. The other piece of choosing an endpoint is, ideally, the endpoint would measure exactly what we want to measure. That is not always the case. Well, it's always not the case, but it's different shades of whether it's the case or not. What we would really like is to measure the true value. That's what we hope to get. But each endpoint also has some level of random error or variance and potentially systematic error or bias. In our clinical trial design, we want to do everything we can to remove as much systematic error as possible. So we can set up our designs and remove bias. It is harder to remove random error. There are ways that we can try to address that, but this really gets to what are you trying to measure, and is the method you have to measure it, how much variability is there in that? For instance, in the pain measure that I mentioned earlier on the scale of 0 to 10, if you ask the same person, within 5 minutes, what's your pain, do you get consistent measures? If you don't, there's variability in that. If you're taking blood pressure, there's some variability in that. The more random error there is, the harder it is to see differences in true values. So the kind of the signal to noise ratio of your endpoint really becomes important for your ability to show effects with reasonable sample sizes as well. I mean, random errors, I mentioned there will be some, you can't ever complete it, but the ways that you can address this is through planning adequate sample size. That's one, but you without kind of increasing your sample size, if you can reduce background noise, control measurement error, it's very common in clinical trials that there's standardized data collection procedures put in place for primary endpoints. If your primary endpoint involves imaging, then there would be standardized protocols that your sites would do within the study. Similar for other types of endpoints with like the Rankin, there would often be training and certification of your investigators to make sure that it's being done as consistently as possible. Ensure data collection is as complete as possible. And the other is really using all collected endpoint data. If you're collecting something repeatedly over time, you don't want to analyze it using just the change from baseline to the end without trying to model all of the time points you have. Because you can reduce your variability by taking into account the correlation among individuals. And the more time points you have, the more you can minimize that error. So that becomes important in thinking of the design as well. And then I think this is a figure many of you have seen in many different areas, but we're looking for both reliability and validity. We may have something that we can measure reliably, but if it's not valid, if it's not hitting the target, it doesn't help us that much. So for instance, on the left, you see a case where we're measuring something very reliably, but it's way off of what we want to detect. That would be a poor measure, as would, all of these others have various levels of problems. What we really want is what you see in the middle, where we're able to accurately measure within some small degree of variability what we truly want to, which is in that scale, that red dot. In terms of choosing endpoints, kind of using Will's theme, although I don't have the pitfall box, although I also played that game as a kid. Choosing endpoints with little known information about validity, reliability, ability to detect change. So if you're using something that's never been tested before, it's really hard to decide what you're seeing. Using some new endpoint without proper pilot testing. It's very dangerous to use an endpoint that's never been used before in your, say, phase 2 clinical trial because the performance of it may not be fully known. The one that is most common, I think, and causes the most confusion is if you're using an endpoint differently than the way it was recommended and validated, that can be problematic as well. I've seen a number of instances where there may be some type of scale that's been validated for use in some disease, and it may be common in clinical trials, and maybe it covers a lot of different scales of disease. You may be mostly interested in any particular scale and want to use the subset of questions related to that scale for your endpoint. If that individual scale has not been validated, that's technically not a validated endpoint. When we validate endpoints like these questionnaires or these scales, they're validated as a whole. So that's something to keep in mind with. Even if you even remove one question from it, it's no longer validated in the way it was developed and proposed. It would have to be revalidated because that one question could affect the performance of this. And validating instruments to collect outcomes is actually a pretty complex and complicated procedure to go through. So that's another thing to keep in mind, particularly if you are in the process of looking at that with respect to your study. We mostly will talk about phase 2 here. We'll kind of introduce this where one of the biggest pieces of phase 2 is this proof of concept, which makes it somewhat difficult. You're not trying to show efficacy. You're trying to really get a sense that there's enough evidence of efficacy and enough evidence of no big problems for safety that it would justify moving forward to an efficacy study. Phase 2 clinical trials are interesting in that I think also this is one area where the rigor of these tends to be somewhat different between industry and academic or NIH-funded trials. One of the key aspects in all, but there's a difference in how it's put together, is this idea of go, no-go criteria. So we're trying to look at proof of concept, and this kind of gets to the point Will had. If you could look to the end of the trial, how would this change what you might do next? And if you think about this, you have some question, you want to address the question. If you're doing the phase 2 and your question is, should we go forward to a phase 3? At the end of the phase 2, when you've collected your data, you decide, do we want to go forward to phase 3? Or does the data tell us it would not be reasonable to go forward to phase 3? And when you're setting up your design, if that trial can't fail, and what I mean by that, if the trial can't give you a no-go criteria, it's probably not a good design. It basically means you've already made your mind up, you're going forward, and you're just going to collect some data to justify moving forward in ways that you've already decided. And that's really not a proper phase 2 trial. The thresholds may be somewhat lower in some industry studies, than they are in NIH-funded trials. Mostly that has to come to if you go forward when you shouldn't, who carries the risk of the future study? If I'm some company and I want to do a phase 2 and I'm willing to have a lower threshold to go to phase 3 and I'm paying for the study, I'm absorbing the risk, that's entirely within, a reasonable decision to make, recognizing the implications. It's a little harder to make that argument if you're proposing for funding from NIH, because it's the NIH dollars, the taxpayer dollars, that are going to absorb that risk. And so there is an idea that if you're going to go forward to a phase 3 trial, you really want to have stronger evidence that it is worth going forward to justify, the taxpayer cost to fund that larger phase 3 clinical trial. There's a number of different types of phase 2 clinical trial designs. I won't get into these very much today, other than to note there's kind of 2 broad types. Ones that use different endpoints than a phase 3 endpoint, and ones that use phase 3 endpoints, but in ways that enable you to answer questions more about the go, no-go perspective than does the drug work or not. So for instance, futility designs are a good example. Futility designs don't tell you a drug works or is ineffective, but it will tell you if the evidence suggests there's not sufficient chance that it would be effective to justify doing the large phase 3 study. A couple of other things I want to hit on just before I close: some common errors in drug development, additional pitfalls, I guess you could say. Poorly designed and optimistically interpreted proof of concept studies. This gets to, again, reiterating the no-go piece. If you can't find no-go, you're going to go forward regardless. It's probably not a good idea. Insufficient exploration of dose, regimen, and route of administration. If you haven't spent sufficient time addressing these things in your early phase studies, then you may fail in phase 3, not because the intervention doesn't work, but because you haven't chosen the optimal dose, regimen, or route of administering the drug. And there's often a tension, and we'd like to get to phase 3 as quick as possible if we think something really works, in order to hopefully show it's effective and get it to patients. But at the same time, if we go too quickly to phase 3, we potentially run into problems where it either doesn't work and would have to be replicated, again, using the example that Will gave, or worse, it looks like it doesn't work and it kind of goes on the shelf, even if it's effective, and then nobody looks at it again because of that study that did not show promise. And that kind of ties in with that next point. The last one, which I bolded, and Will hit on this, and you've heard me say this, but I don't think we can say this enough. If your early phase design looks to be an underpowered phase 3 or efficacy study, that's not only a bad idea, but I think those of us who have been on clinical trial study sections will testify that nothing sucks the air out of a proposal more than having those words uttered. This looks like an underpowered phase 3 clinical trial. That's almost the kiss of death in the review of a clinical trial proposal. So you've heard us say that a lot, but it's mostly because that's a really important thing to keep in mind. There needs to be some clear question and something you would obtain from your early phase design that is beyond, it's just a larger version of what you hope to do in the future with an efficacy trial. Other things that come up with this is sometimes it's not entirely clear maybe what might be the best endpoint, and people are inclined to look at a large number of endpoints, perhaps with the go criteria being if they show a hint of an effect on any of those, they would go forward to phase 3 clinical trials. Just based on statistical properties, it's really easy to show that those are almost surely going to go forward. If I look at 10 endpoints and don't even require statistical significance, just look for a positive trend, I'm probably going to find that either statistically or visually, because I may see something that I like in the data. The other thing is we have to be careful that early phase studies with positive findings, particularly if they're not quantifiable, if I've looked at several things and I'm trying to make a subjective argument, are more likely to be highlighted. That kind of implies that we're more likely to not replicate results than we are to replicate results, because there is some inherent bias, and that we only try to replicate studies that we liked the results initially. Other things are, and this kind of fits into this last point, I guess, that I have here. If it's subjective results, if you don't have quantifiable way to assess your results, you can take, if you have equipoise for a question, someone who thinks the treatment works can look at a set of data and see convincing evidence that it justifies that the intervention works. while simultaneously someone who thinks the intervention does not work can look at that same set of data and see convincing evidence that it does not work. We can see patterns that are consistent with kind of our inherent beliefs, and you want to try to avoid that by putting that in. The last thing I have, which I think is an important consideration, I'm not going to go too deep into this because that's what our webinar next week is on, is sample size. And really getting all of this in place with your endpoint, your objectives, your design, is enable you to do a valid sample size calculation. And for those of us who are statisticians, when we work with clinicians, they, for the most part, trust us with writing the statistical analysis plan and the analysis sections. As long as it passes muster, they're not going to worry too much about what the wording is or what we're proposing, as long as it gets past those who would review the protocol. Clinical investigators will, and you should, those of you doing this, care a whole lot about what your statistician brings back with the sample size calculation, because that has major implications on your study. Most notably, it's going to be one of the biggest budget drivers, because the sample size calculation is going to tell you how many participants you need to enroll. And obviously, the more participants you need to enroll, the costlier, and both in terms of money and time, that a study may get. The reason why you have to address these questions early is that it has to be consistent with all of the following: the aims, the objectives, the design, the endpoint, and the statistical analysis plan. So by this, I mean, you want to have your sample size calculation match what you're proposing to do. If you are going to do a chi-square test for your primary endpoint, you don't want to use some t-test, as an extreme example, to justify your sample size. It should be justified based on the same set of analyses that you're going to do. And I just put this as last just to show that, and I think this will come back when Sharon gives her presentation next week, but you can't really separate power from the size of the study or the magnitude of the effect. They all three have to be given. Statements like this, which are they've been slightly changed, but represent statements that I have actually seen in protocols in the past. The trial has 90% power. Every trial has 90% power for something. It depends upon what effect. Even this, which gives some, will have 80% power to detect meaningful differences between the two groups with 100 individuals per group using a two-sided type 1 error rate of 0.05. That sounds good, but it never tells me what a meaningful difference is, right? What might be meaningful to you might not be meaningful to someone else, so I have no way to judge that. And then even below, we'll have 88% power or greater to detect a relative risk of 1.5 or greater for all biomarkers using a two-tailed type 1 error rate of 0.05. That's still a little vague in terms of what we're proposing to do or what we would be able to see, in the sense of a relative risk of 1.5 without kind of knowing what the underlying risk in the control group is a little hard to justify. So this is kind of feeding into what I hope will be something that you'll think about as you come into next week's webinar, and think about what are the pieces that you need to have in order to give the statistician to do the sample size, but also to write the appropriate language and make sure you have the pieces in your protocol to replicate that. And I will pause there, and I think Will or I can take questions. Yeah, Will's comment is probably the biggest detriment to having a large number of secondary objectives. If you have 50 secondary objectives, you have to enter them into clinicaltrials.gov.
Another thing about phase 2 trials, and I can sort of, I mean, anything you do where you look back at a phase 2 trial and then the phase 3 trial was neutral must be taken with a shaker of salt, because you're sort of going back and dredging. But one example from our own, sort of kind of airing my research networks and dirty laundry, we did a study of progesterone and TBI, a phase 3 study in the Neurologic Emergencies Treatment Trials Network. And the phase 2 trial that led to the enthusiasm for this showed a really marked decrease in mortality. The mortality in the placebo group was 40%, and it was 13% in severe TBI and 16% in moderate TBI. So that was, you look at that and you're like, that's really promising. But then Chris looks at it and is like, you only had 23 people in the placebo group. So this 40% is a very noisy estimate. It looks like mortality. Here also you see something that's unexpected in that there's less mortality in the people with severe TBI. So this is a signal of something as a problem. And then moderate. Possibly in retrospect, this was the fact that sometimes people who are drinking or doing other drugs have TBI, and they tend to have a depressed GCS, possibly. But for an emergency trial with a short treatment window, they need to be put in the trial soon. But the thing that's probably most important is even though they had sort of, if you're picking mortality as your main outcome to sort of say, we should progress, it's really telling that for the, severe group, when they looked at this as dichotomized based on the Glasgow outcome scale, there was a really wide confidence interval in this treatment effect. So if they had go, no-go criteria on this, they wouldn't have moved on to a phase 3 trial in all likelihood. But everybody was so, and again, there's more to this story than this, but it's just one of those things that if you do have, if you do have a main outcome, but you have some secondary outcomes that ought to be going in a similar direction and they aren't, it's important to think about those, since you have collected, you want to be able to think about those and how they may be giving you a signal of something that could be important and should give you pause as you design the next phase. I'll put a PubMed link in for that article so that people are like, he's citing some random thing. And now, I mean, I can go on a little bit of the soapbox. One of the other interesting things about that is animal data suggests that we needed to treat people with progesterone for 5 days, but all the clinicians got in the room and sort of told the basic scientists, like, we can't run a progesterone infusion for 5 days, and the clinicians like, why don't we just do 2? And had there been a larger pushback and had it been studied at a longer dose, could the entire sort of research enterprise gone in a different direction? We don't know because there's not much enthusiasm for retesting that agent because they elected to go with that shorter dose. Like when I said you make a bunch of these assumptions at the beginning, one could say, okay, when the patients start to wake up, we'll start giving them progesterone pills. But, that's a slightly different compound. So there, you get into these practical issues of like, well, the patients need to move on to rehab. We can't run an infusion for that long. And there's too much messiness for transitioning somebody to oral drugs after IV. So they were left with maybe that, with this regret, a number of years later of if we had treated the patients longer, maybe we would have seen something different. So those are sort of real life examples.
Yeah, I think sometimes that's one of the things that you kind of struggle with is dose and duration. And if there's not really clear data, how do you choose dose and duration especially in a rare disease where it may be difficult to, but that's a whole nother, talk, right?
I think it also
Well, I see
gets some to the, like, trying to leave our preconceived notions out of this, and that I think, I mean, one of the things I tell all the statistician students that we have in class here is your work is not symmetric based on the results of the study. If the investigator gets what they wanted out of the study, they're ready to go and write the paper. When they don't, you end up doing a ton of post hoc exploratory analysis trying to decide why what I thought was going to work did not happen. So, right, if there's a fluky thing in the positive, we don't break it down and say, well, this looks too good to be true. Let's dig into this and see why it's too good to be true. That is rarely done.
So one thing that we do sometimes when we're working with people who have a promising agent, when we're working to help them with a trial design, is we try to get into that via anticipated regret.
Right.
So we say like at the end of your trial, let's say the treatment only works in women. Do you have what would you be sad? Or like, no, that would be great. I would run a trial. I was like, well, do you have a biological hypothesis for that? They'd be like, sure, this is the biological hypothesis for that. And we're like, okay, what about if you ran your trial at the end, it was shown to only work in men? And they're like, oh, that would be great. It's like, do you have a biological hypothesis for that too? They're like, oh yeah, it could, and you're like, okay. So if it was sort of like anything that they would see, they could construct a biological hypothesis for what it was. So I think being careful about how you pre-specify how you might use that information is really important. So you don't go, searching. Anyway, I see like Melissa and Sherry might have questions, so you should.
Melissa, did you have a question?
Sure. Yeah. Thank you both. That was very helpful. Even post 24 hours call, learning things. I know, Chris, we talked a little bit about this in our small group a week ago. I'm kind of curious, both your and Will's thoughts. Slightly different philosophical question, but thinking about choosing primary objective and endpoint time-wise and sort of the proximity to the intervention, my personal interest in cardiac arrest, a little less dose and timing in intervention there, but of course a disease where we expect to see over time continued recovery. And looking at our RCTs of ECPR and out of hospital arrest, we've got a wide range of primary objectives or primary endpoints that have been used historically with varying results. So I'm curious how you, and in critical care studies in general, is it survival to discharge, 30 days, 180 days, and see what that recovery looks like farther on where it's less proximal to your actual intervention.
Maybe I'll let Will take that first since you've heard some of my thoughts on it already, and then I can weigh in.
I think probably the field is chastened somewhat because of prior things that sort of went in a not so ideal direction, in that, like, if we look at short-term mortality, then we learn, oh, all these people, not all, a large portion of these people are neurologically devastated. On the flip side, using the completely other approach of like, people may be waking up to a year, we should be making, we should use a year. Then that may allow people longer to wake up, but it also allows all sorts of other bad things to happen to people in between that were unrelated to the proximity of your intervention. So I think it's tricky. I think there's never one size fits all question. I do feel like there is, I think there in this setting, I think there's potential of other sort of surrogates, like, time to awakening and things like that could be more proximate to your initial intervention and ought to give you a sense as to the preliminary effect. But that's been a challenge in both stroke and cardiac arrest research, is getting a more proximate outcome that is reliably predictive of the long-term outcome. Even though there's a lot of things that are promising, they all have a little bit of baggage, and I think you just have to be cautious as to how you use them and justify them. But I agree that usually there's so many other things that happen in these sick patients if you use too long a duration, that it's important, but it's also, it can also be a bit of a problem.
And we'll continue in the group, but I think this is not just a problem for the example like what you're talking about. Even with more like standard type clinical trials in a lot of rare diseases, it may not be clear what the endpoint is. And really, to succeed in developing a new intervention or a new drug, you have to have an effective drug and you have to have a good endpoint to show that the drug is effective. And if you don't have both, it really complicates it. And it's not quite the same problem you're raising, but it is a challenge anywhere where you don't have kind of a for lack of a better term, plug and play endpoint that's been vetted and known to be a good, indicator of efficacy. I think we're almost out of time. I think, Will, you said Sherry might have a question. Sherry, did you have a question? Oh, okay. Okay. I think if there's no more pressing questions, we can turn it back to you, Megan.
I have one question. Sorry, I'm late.
Sure.
Turn on the camera. So should you power your study to look at both primary, secondary, and exploratory aims?
Well, there's different schools of thought on that, I think, and you're talking, and there's a difference, I think, between phase 3 and phase 2 studies as well, right? And this is partly why. So in the course, we're talking more in the phase 2 and early phase trials, and I would argue there you're really mostly powering it for your main question, which is where you're go no go. And part of the reason why I say you should limit your number of secondaries, in my experience in phase 2 clinical trials, there's not a big difference between something somebody calls a secondary endpoint and exploratory endpoint. They're usually not powered, almost by definition, because of the sample size you're talking about. So I think the ultimate is for phase 1, phase 2 studies is powering it for your main question. When you get to phase 3, that's where you may want to make sure you have some level of power for your secondaries, in a regulatory trial, particularly because that's going to help support it and give the totality of evidence. So it's a little bit different, I think, based on where you are in the space. I don't know, Will, if you have thoughts.
Yeah, I will also say that some grant reviewers are not going to be as sophisticated. So if you are doing an early phase, say R01, and you name things as secondary outcomes or secondary endpoints, but you do not show what power you have for them, that they will ding you for it. But I think Chris's philosophy is correct. If you're doing a proof of concept or early phase study, you're really, and you're just sort of, like say you're doing a stroke study in 10 patients, you're collecting MRS, but you really should be saying explicitly, we're going to give you the proportions and you can take them as you will. We should not do a hypothesis test on that. Some people will sort of say, if there isn't a hypothesis test, it ought to be an exploratory outcome. And I think saying you're going to do effect size estimation or just give confidence intervals on some of those non-main outcomes is a scientifically transparent approach. But depending on what sort of type of submission you're doing in terms of getting funding, you may have to say, oh yeah, we're going to look at mortality, but we have this much power to show a difference in mortality, which is going to be less for your small stuff.
I think also that gets into in proposal a very different world in the way people write. I know we're a bit over, so I want to make this one last point before that. In industry studies, each clinical trial is part of a larger program, and it's kind of clear where each fits. We don't think that way when we submit NIH grants as much, but I think part of like this problem, and even with Will talks about if you give things, if you can explain what is potential next steps, like especially if it goes forward, what is the next step and how all these pieces will inform it, that goes a long way for addressing some of these questions. We don't think that way often when we submit grants to NIH for clinical trials, but especially for these early phase trials, there should be some thought of, if this is successful, where does it go next? And how do all of these pieces of information give me what I would need to do that? And I think if you can tell that story, it won't solve it in terms of the reviewer problem Will mentions, but it helps because then you can kind of see why knowing this helps give me a sense of what that next study would be and how it all flows together. So it kind of as a broad answer to your question, but more highly, I would think everybody should be thinking of, if you're doing an early phase trial, which almost everyone in the course is, where could this go from here, right? And the thing that really becomes a problem in review, even if it's a great study, a great idea, and it's not clear that it would ever go anywhere after that, that's going to be a problem in terms of, are you going to get funding to do that study? So you should always be thinking about where to go, as your next step. I know we ran over, Megan, so I'll turn it over to you.
Okay. Thank you everyone for joining us. Please grab the evaluation link in the chat before you go. And you can learn more about sample size next Friday during our next webinar, which will be presented by Sharon Yates. So we hope to see you there. Have a great weekend.
Thanks, Will and Chris.
Thank you.